I will talk today about the design of repeat proteins as recognition elements for microbial sensors. Infectious and parasitic diseases are a second leading cause of death worldwide, according to the CDC and World Health Organizations. And numbers are especially alarming when we talk about developing countries. Traditionally, detection techniques for infections and parasitic diseases rely on cell culture assays, microscopy, and biochemistry. And although all these techniques work really good and are very specific, the problem is that they usually take a long time, sometimes over a course of several days, and requires a lot of the cell handling. This is usually okay when we talk about typical infections like a strep throat that many of you have experienced. However, the time between diagnosis and treatment becomes very important in some serious diseases like meningitis or food poisoning or bioterrorism. In those cases, we really want diagnosis to happen very quickly so that the treatment can start in a course of several minutes to hours rather than several days. And in these dire cases and scenarios is where standalone biosensors that can be applied as a point-of-care diagnosis devices are becoming increasingly important. When we talk about biosensors in general, we talk about architecture that has a recognition module, and so this will give specificity to the biosensors and decide what we are really sensing, and then a transducer element that is really a physical technique that will allow us to read out recognition of the desired analyte. And the transducers can be optical element, electrochemical, piezoelectric, magnetic, but really what we are mostly interested in this talk is going to be design of a recognition element. As I said, that one of the goals for our biosensors is that we want it to be rapid, sensitive, selective, and simple so that it can be really used by anybody at a point of infection and sometimes in a field. When we talk about bacterial and microbial sensing, we also need to keep in mind that a lot of sensing that we do is going to be happening in a very complex matrix. So we are talking about in a complex matrix of analytes, for example, testing the waters or testing the soil, or in a complex body fluids such as saliva or blood or urine and so forth. So if we look at the bacterial architecture and think what can be good analytes to sense with the biosensors, we can think about specific bacterial proteins, DNA, and RNA, and there have been many sensors that are listed here in the references that have been recently developed and that work actually very successfully. However, one of the problems with it is that all of these sensing of things that are happening inside of the bacteria also requires handling of the cell, unless we are talking about excretion proteins. And also, when we talk about the really good bacterial biosensors, we ideally want to sense something that is essential for bacterial life because bacteria multiply and mutate much faster than we can do the science. So we are talking about something that is going to be essential for bacterial life and something that is not mutating fast, something that is, in a sense, evolutionarily conserved. And so the other aspect that we want to pay attention to is if we want to minimize the handling of bacterial cells, is if we can do the sensing in the context of the whole cell. So we don't need to lyse cells and we don't need to repurify and isolate analytes, but really just sense the cell, in a sense, cell surface. And so thinking about that format of sensing, we can start thinking about what is in a bacterial cell wall that can be a good analyte. And one of the things that my lab is interested in is looking at the bacterial peptidoglycans. All bacteria have fairly thick peptidoglycan layer on their surface, where in mycobacteria, additionally, that peptidoglycan layer has a chemical modification that is glycosylation, and I'm going to point out that modification later because we are interested in that one. So thinking about all these requirements for a biosensor, we went back to the nature to see how is nature solving that problem, because nature obviously has mechanisms of fighting bacterial infection every day. And one of the proteins that have a role of first responders are innate immunity receptors. And we were especially interested in a group of innate immunity receptors. These are cellular proteins that are expressed in the macrophages that are called NLRs. So if you look at the NLR architecture, it's actually very similar to the man-made biosensors. We have a recognition module that is composed of the tandem leucine-rich repeats. And then we have a nucleotide binding domain has a function of the transducer element, where upon recognition of the analyte, these proteins will either oligomerize or interact with the caspases to signal downstream inflammation. And so we were really interested in this natural way of biosensing, and we hypothesized that we can redesign leucine-rich repeat domains from these proteins to help us design the biosensors for bacterial cell wall. So here I will present you design of the consensus sequence of the subgroup of NLR proteins called the nodes. And these node proteins are characterized with the LRR domains that are 28 amino acids long and occurred in a tandem anywhere between 3 tandem repeats all the way to over 20 tandem repeats. We have analyzed node subfamily from mammalian organisms. And so I will quickly now show you what the sequence analysis tells us. So we have analyzed the N-terminal internal repeat and C-terminal repeat separately. And the conclusions that we can draw from this analysis is on the top row, we are looking at the amino acid identity in each table. And in the bottom row of each table, we are looking at conservation of physical properties of amino acids. And so two conclusions that we can make is that N-terminal repeat and C-terminal repeat have overall higher conservation than internal repeat, but also that the physical properties in all 3 kinds of repeats are much more conserved than the actual amino acid identity. So with that in mind, we went and we have made the protein that we termed CLRR2, where C stands for consensus, in which we have 1 N-terminal repeat, then 2 internal repeats, and then C-terminal repeat. This protein is well expressed, it's folded, it has a TM of about 60 degrees Celsius, and to our actually very big surprise, this protein is capable of binding fragment of the bacterial cell wall called muramyl dipeptide without us performing any special affinity maturation. So we have performed binding assay looking at the fluorescent quenching because in the protein sequence there are 3 tryptophans. And we can also do this reversely when we can label muramyl dipeptide with the fluorescent tag and then look at the fluorescence and isotropy upon binding of the protein. This was important to do experiment in both ways and confirm the binding because we did not engineer affinity and we just wanted to make sure that what we are detecting is not some non-specific interaction. To our even bigger surprise, this protein not only binds bacterial cell wall, but it's also very selective to the physiologically important form of muramyl dipeptide. So CLR2 has a 2 micromolar binding to LD-MDP, which is physiologically relevant form of muramyl dipeptide. However, it does not bind optical isomer, DD-MDP, or it does not bind this glycosylated form of the MDP that I mentioned is present in the mycobacteria. So in conclusion, we have used the consensus sequence of LRR domain of the NOD family of innate immune receptors to create stable monomeric and cysteine-free scaffold that without any affinity maturation has a micromolar binding to muramyl dipeptide bacterial cell wall. CLRR2 also distinguishes between structurally related and similar ligands, indicated that this might be a good starting scaffold to start making binders for other important ligands because we can obviously impart selectivity and sensitivity between structurally related molecules.